Hey there, we are going to review enzymes today. We're going to start off talking about the other name that enzymes are known as is a catalyst, which are a special type of protein, right? One of our organic molecules. Therefore, like all proteins, enzymes are made by ribosomes. Remember, we learned about ribosomes back in Unit 1. Ribosomes make proteins. Enzymes are a specific example of a protein. The function of all enzymes is to speed up chemical reactions, and they do this by binding to the substrate, and the whole process looks like this. So this would be our substrate. It would bind to the enzyme, right? That's what this whole thing is. And the place where the substrate and the enzyme bind, right, that is the active site, right? The part of the enzyme that touches the substrate. So the substrate and the enzyme bind. It creates what's called the substrate enzyme complex, and then that's when the chemical reaction occurs to create products. Now, enzymes work by lowering the amount of energy needed for this chemical reaction to occur. So we are lowering the activation energy, right? Which means less energy is required to get the reaction started. So enzymes basically make it cheaper for our body to run these chemical reactions, right? To take in a carbohydrate, like let's say lactase, which is a complex carb, and break it down into simple carbs, or the monomers, glucose. So enzymes speed that reaction up. They make it go faster. But enzymes cannot work if they are denatured, right? They're supposed to function forever and forever and keep being reused, but they can be denatured by things like temperature, pH, and salinity, or basically the amount of salt that's in our system. And because enzymes only bind to one particular substrate whose shape matches the enzyme's active site, we call this the lock and key model, right? For every substrate, there is a matching enzyme. So that's it for our enzyme review. If you have any questions still about enzymes, please be sure to let me know.